So good afternoon, everyone. I'm very happy to be here. And first of all, I want to thank Maritoni, Alicia, and Paco for inviting me to share with you the work I've been doing recently with Pepe. And I have to give all the credit to David, who in fact designed everything in the story I will tell you about. So before we start really to talk about the epigenic biosensors that actually David designed and processed, I just want to highlight a little bit what we've been doing in the lab over the five or 10 years before. So we are interested in phages, whatever phage means. Prophages, phage outside. We really like to understand how phage and the host can talk to each other through genetic cross-regulation, and actually how prophage encoded regulator can integrate into the host regulatory network. We are also interested in host, how does it work? In host takeover and the early infection steps. And also I will talk a little bit about the phage, how phage and antibiotic can synergize under certain conditions. We are also interested in phage diversity and genomics, and I will display a little bit of the results we had with phages isolated here in Sevilla. And I will talk mostly about biosensors, but from two sides. First, I will talk about biosensors that we designed before I came to Sevilla. Those biosensors are phage-based and are used to detect and count bacteria in the environment. And you will see how David imagined that we can use bacteria and bistable expressed operon to detect and to enrich phages from the environment. So the other way to look at it. So you probably all know about how phage are wonderful nanostructure and nanoscale tools. And many of us, of course, heard about phage display. But by engineering phage, you can use them as imaging agents, either using filamentous phages or icosahedral phages. You can use them to produce multifunctional probes by covering the capsid, whatever the capsid is, with different epitopes or functionalized protein. And you can also do target immobilization, signal amplification, because these are very easy to produce and you can accumulate them on surfaces, and of course to detect bacteria. So why use bacteriophages as biosensors? Well, they specifically infect bacteria. They have variable host range. Most have very narrow host range. So it means that you can detect specifically one type of bacteria or one subtype of bacteria. But they can also be detect or infect cross species. So depending on the nature, either virulent, meaning that they have a lytic lifestyle, obligatory lytic lifestyle, or temperate, you can design differently your biosensors. So we'll talk about temperate phage-based biosensors, because when we started that kind of project, it was easier to engineer temperate phages than a lytic one. And of course, one of the advantages that they are so abundant. You can go in any pond, any kind of environment, and you can dig and sample and get your phages against the bacteria you want to use. Of course, you have to choose the specific environment you want to study and the bacteria you want to study. And I will tell you a little secret that we had about Sevilla. So I like to to play between basic science and how we can understand and how we can develop specific strategies to understand host-phage interactions, but also we like to play with applications. So here is an example of things we've been developing very recently, which is real-time infection tracking under the microscope. And what you see is that you have a mix of resistant and sensitive strain. And what you see is that the epidemics can progress into a lot of mixed bacteria and still happily living whenever they are resistant. So this is an example of spatial spreading of an epidemic. And of course, physicists are very excited about this kind of development because they can model the epidemic, the spatial way of development of the epidemics. And I can tell you that it's a bit easier than to do it with humans, of course. So we are currently designing a new model for the spatial spreading of those kind of epidemics in mixed culture when you have resistant, when you have sensible bacteria. But it also allows you to do real-time experimental evolution, because if you look closely, here you have one green cell remaining, and this one cell will further develop. So you can play with evolution in real time, with viral adaptation in real time. So Julian Burcico, and you can see by the accessories that he's from Argentina, he developed very powerful tools, and in particular, he was able to label some phage. So this is the HK620 phage, which is very closely related to P22 in Salmonella. And so you can look at the number of phage binding to each bacterium. So the idea is that his project is really about looking at the synergy between phages and antibiotic, and he was able to count individually. So we can do single cell, but also single phage study. He was able to look individually to cells, either untreated or treated with cephalexin or ciprofloxacin, where you can see that there is a very important filamentation effect of those antibiotics. And the idea behind those images is that the longer the bacteria are, the more phage attached to them. And in fact, the synergy is as simple as that, because you have a huge cytoplasm that allows the production of a higher amount of bacteriophages. So let's go back to the biosensors. So the first biosensors we designed, as I told you, were based on temperate bacteriophages, because it was very easy to engineer the bacteriophage when the genome is integrated into the host genome. And then if you can do genetic, like in E. coli or in Salmonella, it's very easy to engineer your bacteriophage. So the idea is that by adding a GFP gene fusion, upon infection, the phage injects its DNA, and then your bacteria grow. So it's very easy to analyze and very robust to analyze using flow cytometry. And of course, you can do single cell analysis and analyze other parameters such as the size, the shape, and the level of fluorescence. So at that time, Manon Vinet and Nathalie Franche developed these biosensors in the lab. And for example, this is a biosensor based on P22 that carries a PRRNB GFP fusion. And you can see that we can discriminate very easily the infected cells from the non-infected cells. But so this is pretty easy. You have a pure culture, you design a biosensor, and yes, you detect almost 100% of the bacteria. Fine. But what happens when you mix the bacteria? And this is the example that we had with mixing E. coli. So you have one E. coli cell for 9 Salmonella cells. And you can see that using an E. coli biosensor, you can discriminate exactly 10%, almost exactly 10% of the cells that are fluorescent. So meaning that in mixed culture, it works very well. So what we've been designing was a fast and specific detection using phage to discriminate bacteria. And actually, it was very fast as well because it was less than 4 hours from sample collection to specific detection. So we had versatile versions from E. coli and Salmonella. And also, it came in various flavors because instead of a fluorescence gene, you can insert a luminescence operon. And that gives you something which is easier to use in the field. Because then the idea was, of course, to test directly in the environment. And it was really our proof of concept to test it directly into the seawater, where you have many other microorganisms. And it was very easy to detect Salmonella and E. coli in such environment. So this is almost where I was when I came, and I had the great pleasure to do a sabbatical stay in Pepe's lab. So it was 5 years ago already. And in addition to what Javier said this morning, an important lesson I got from Pepe was that not only was giving a strong mentoring, but also he was giving freedom to the students. And to me, it was really a huge lesson, because when I met David, he was the one who asked the question. He was the one who designed the research he wanted to do. And the objectives of his PhD was actually to use an epigenetic fusion to do whatever biotechnological tool. So he came to me, and I was discussing at that time with all the people in Pepe's lab. And he came to me and he was asking this very straightforward question: can we design a phage biosensor using an epigenetic reporter? And I was like, yeah, why not? Let's try. So the idea of David was actually based on a former work done by Nacho Cota in Pepe's lab. And what Nacho observed was that, so he was studying the expression of the famous OPBAB operon, and I think Maritoni will talk about more later on. And the thing is that when you look at the LPS, what Nacho found is that the expression of OPBAB does not modify the nature of the LPS, but the length of the O antigen. And because the expression of OPBAB is bistable, you have a mix between the off subpopulation with a full-length LPS and the on subpopulation with very short LPS. So in the absence of phage, you have a variable proportion depending on the cell cycle, depending on the methylation of the promoter. So you have heterogeneous expression of OPBAB operon. But what Nacho showed was that in the presence of phage, you actually enriched one subpopulation, the one that resists to the phage. And in fact, it was the OPBAB on subpopulation. So the idea of David was very simple, but in fact very efficient. That if you use an OPBAB fusion with the GFP, and you add phage, you enrich the on subpopulation, and then you can measure the number of phage. You can have an indirect measurement of the number of phage in the solution you have. So first, he did a proof of concept with known phages, such as P22, a virulent version, which is called P22H5, but also DET7 and 9AA. So what you can see is that using flow cytometry, there is a very strong enrichment of the on subpopulation in the presence of those bacteriophages. So the next step was to go and pick samples everywhere in Sevilla, and mostly in the ponds of Maria Luisa Parque or under the bridge of Triana. And so the idea was to isolate new phages. So you have this prefix here of these beautiful nanomachines, and the prefix SE is for Sevilla, of course. So what he obtained was a bunch of phages. We didn't analyze them all yet because there are many. And you can see that all of them, these new phages, can enrich, but not to the same proportion. They enrich the on subpopulation. So for example, this one, the SEML, you would say, okay, the enrichment is not so good. So it's very informative indeed, because remember, the OPBAB operon changes the length of the LPS. So it means that in this case, you may have a secondary receptor for the phage, which is very important also to know. So next, he studied the sensitivity of the detection. By doing serial dilutions, he was able to show that, well, he could detect around 100 phage plaque forming units per ml in a plate reader, and using flow cytometry increased a little bit the detection limit to 10 PFUs per ml. Well, it was good, but not enough for David. So the next step was to improve the sensitivity. And because of the properties of the OPBAB operant expression, he knew that by mutating the GATC site, he could improve the expression of the GFP fused to the promoter. So in that case, he also showed that if you lower the number of phage at the beginning of the infection, in fact, the amplitude of the detection was even more important. So by doing both, decreasing the MOI at the beginning of the infection, and also by using a fusion where the GATC1 and 2 sites are mutated, he was able to lower the detection limit to 8 PFU per 500 milliliters, which is about 15 per liter, which is extremely low. So he was very happy, but not yet finished. The next step, he was like, okay, so now I know how to design a biosensor to detect Salmonella phages, but let's do it in E. coli. So in E. coli, even though the LPS is very important, it's probably less important than in Salmonella for phage binding and phage infection. In E. coli, you have much more proteins, outer membrane proteins involved. And actually, he decided to design a biosensor of phages that were using FA2A here, which is a siderophore receptor protein that allows bacteria to capture iron, which is also the receptor of several bacteriophages such as T1, Phe80, and T5. But so when he started to think about this, he thought that, yeah, okay, if I'm doing the POPBAV few A GFP fusion, I will look at a decrease of fluorescence. And this is not the easier when you want to detect tiny amounts of something into the environment. So he said, no, let's change that and use the fusion with a protein that inhibits, in fact, the binding to this receptor. And he did the fusion with OPBAB LLP GFP. And using this fusion, upon phage binding, you have an increase in fluorescence. So this is what he did. And here is the control with a non-phage T5. So you can see that there is a clear increase of the subpopulation that express the inhibitor. And so you can detect phages that bind actually to few A. But also, he isolated new phage from against E. coli. Oh, and this is an interesting control as well, which is the use of the colicin AIM, which is also binding to the same receptor. And you can see that colicin can also be detected through this biosensor. So the idea is that by using different gene fusion, you can not only detect the bacteriophage, you can isolate the bacteriophage you want to pick. But one thing which is very important, and especially for phage therapy, is that you need to know the receptor. And the sequencing won't give you the receptor the phage is using, in fact. You can sequence whatever you want, you will never know exactly what is the receptor. By using this kind of experiment, you can discriminate between receptor, and for designing the cocktails for therapy, in fact, you need to mix phages that use different receptors, so you don't get the whole pressure, evolutionary pressure on a single receptor. So this is almost the end of the story of these epigenetic biosensors. So finally, we got a very good specific detection that helped to isolate in less than 24 hours from sample collection to specific detection. And the most important, it's without any enrichment step. You don't need to enrich your sample. So we have E. coli and Salmonella version. You can discriminate, as I told you, between different receptors. And the trick I was talking about is that you need a place where you have ducts, because if you have ducts, you have Salmonella. If you have Salmonella, you have phages. So the phages, the SE phages for Sevilla, we went on studying them because there is a huge demand for those phages, and in particular for biocontrol in the plant fields, and also in the chicken industry, in food industry. So we sequenced and analyzed all these brand new phages. We were able to attribute new families, and in particular, one brand new temperate phage, because temperate phages from Salmonella are very rare, finally, although we have P22, but it's almost the exception. So we got a brand new temperate phage, and we were very happy about this. And incidentally, this publication got accepted just last August. So finally, I want to thank the people and all the people who really welcomed me here in Sevilla. Here is one of the occasions we had a very good lunch. I want to thank, of course, David and Pepe to have shared these results with me, to have pushed me to the limit of the detection, and of course, all the ducks of Sevilla area. I also want to thank all the Phage aficionados from Marseille and my group with the great support I have from them. So I mentioned Julian for the real-time analysis of phage infection, but also I want to thank Nicolas Ginet, who is our genomic expert in the lab. And finally, I want to share with you these good memories. And the last time I've been here in Sevilla was exactly 3 years ago, and it was for David Defense in the building next door. And as usually, because we spent such a very good time, actually Pepe was a bit joking, making jokes of me, and because he was saying that I was a best expert in the Triana Mercado than himself. So we loved our Triana experience with the whole family. And this is a picture that we took in Carmona, where we had, as usually, a wonderful lunch together with Pepe and Laura. Thank you for your attention.
Thanks so much, Mireille.